The overall goal of this procedure is to explore gene function in Dermestes maculatus, a new insect model system, which represents the diverse clade of beetles and facilitates the study of basic and applied research questions.
This method can help answer key questions in the fields of developmental biology, evolutionary biology, and beyond by making it possible to test gene function in Dermestes embryos and adults. The main advantage of this technique is that RNA interference can be used to target any gene based only on sequence information, even in the absence of a full genome sequence.
Methods introduced here not only provide an approach to study gene function in Dermestes, they can also be applied to develop new insect model systems and potentially to control insect pests.
To prepare a rearing cage for D. maculatus, first spread a thin layer of wood shavings into a medium-sized insect cage. Place a section of styrofoam into the cage to allow larvae an environment in which to pupate. Add 20 to 50 adult beetles or late instar larvae to the cage. Place wet cat food into a petri dish or weigh boat as a food source. Cover the cage with a mesh cloth and place it into an incubator. Before starting embryo collection, first check the cage carefully for any embryos present and remove them. Once the cage is clear, place a stretched cotton ball into the cage and incubate the colony at 30 degrees Celsius. After the appropriate